Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjon classes. Dear students, we are initiating today a new chapter of class 12th, that is strategies for enhancement in food production. Dear students, the name and suggest we are going to talk about some of the strategies which are actually helpful and enhancing the food production. And we know that humans derive their nutrition from plant and animals. So in this chapter, two parts we discuss actually, that is animal husbandry and the plant breeding. Animal husbandry will be covered in the zoology part in a separate lecture, but here today we are going to discuss about plant breeding in detail. Okay, so let's discuss about the plant breeding. So first thing is what is why we need to discuss about this chapter. So as I said, the human derived that nutrition from the plant and animals. Initially, we know that human hunted wild animals and collected fruits from the wild plants. So initially, it was happened that hunting was for the food purpose, the collection of fruit from the plant for the food purpose. But much later, different organisms begin or they begin in humans begin to cultivate plant species and rare animals under their supervision. We know that as human population is rising steadily, the requirement of food is rising proportionally. So as the human population is increasing, the requirement of food is also increasing proportionally. Therefore, there is a need of continuous enhancement of food production, and that has become a major necessity. Animal husbandry and plant breeding have to play a major role with to this purpose to enhance the practices of food production. So today, with respect to plant, how we can breed a plant, how we can increase a yield, how we can increase a production of a food, that we are going to discuss in this plant breeding chapter. So traditionally, dear students, farming can only yield a limited biomass. We know that. Yes or no? And that limited biomass for the as a food for the humans and animals. And this limited biomass, it is not possible to provide to all population or the increasing number of organisms. So there are a need of better management practices, and that increased at the course of duration, and increases in average can increase yield. Okay, so better maintenance practices and increase in average in the crop yield. So increase in what a crop yield, but only to limited extent. So we know that the better maintenance practices increase the crop yield, but only up to a limited extent, which was insufficient to meet the requirement of rapidly growing human population. And if we talk about average area of land measured in mass, where the proper, we can say, the average production was increased, but it is insufficient to meet the requirement of rapidly growing human population. In India, if we talk about green revolution was responsible for our country to not only meet the nutritional requirement of food production, but also help us to export it. And green revolution was dependent mainly on plant breeding technique for developing a high yielding and disease resistant varieties in wheat, rice, and maize. So plant breeding is the one of the important technique which helped to increase the yield to a very large extent. And the green revolution happened, and that due to the breeding techniques for the development of high yielding and disease resistant varieties of wheat and maize and rice. So who is in India has not heard of the green revolution? Everyone has heard, and that is related to the what we can say. the agriculture. So green revolution made it possible for our country to meet the national requirement in food production. And the green revolution refers to a series of efforts. Now, what is green revolution? If you answer a question, so it includes a series of research development, technology transfer initiative to the different countries occurred between the 1940s and late 1960s. And due to this technology transfer initiative, research in the field of agriculture that increased the agriculture production worldwide, particularly in the developing world or developing countries, significantly in the late 1960s. So that is what a green revolution that increased the yield, increased the production. So here the major focus is to produce the improved varieties, and that improved varieties which will be going to produce by this plant breeding will have a better yield, better disease resistant, so better production can be available. So it is known that Norman Borlaug is considered as the father of green revolution. Okay, and he is a Borlaug. Now, green evolution, if we talk, was dependent to a large extent on plant breeding technique and the development of high yielding and disease resistant varieties. If we talk an example of such varieties whose disease resistance and yield was improved, that is wheat, rice, and maize. So this is the wheat, this is the rice, and this is the maize, because it was used for a long period of time as a major crop. Okay. So now the question is, we are talking till now that breeding we will do, and breeding will increase the yield, increase the production of disease resistant plants. So the question is, what is plant breeding? What is this breeding? So if we answer this question, plant breeding is a purposeful manipulation of plant species. We are going to manipulate it. We are going to modify the plant species, and that is in order to create desired plant types. What we are going to create, a desired plant means whatever the particular characteristic we need in a upcoming new variety. Based on that, we carry out a purposeful manipulation, and such a way the desired characteristics are obtained in the new variety. And this new plant with the desired characteristic are better suited for cultivation. They will give the better yield, and they will be obvious disease resistance also. So this is a plant breeding, and conventional plant breeding has practiced for thousands of years. Conventional is traditional plant breeding, and this is a what a particularly we can say the practiced for the thousands of years since the beginning of human civilization, and record evidence of a plant breeding dates back 9,000 to 11,000 years, means such plant breeding was done before 9,000 to 11,000 years. Many present day crop, if we talk, they are the result of domestication in ancient times. Okay, so there are many crops, such crops are seen. Okay, so bringing wild varieties under human management is called domestication. So what is domestication? So domestication is the process of bringing a species under human management. Today, all our major crop are derived from the domesticated varieties, which are marketed in the market, which are in the human management. Classical plant breeding involves the crossing and hybridization of pure line. Pure line means homozygous plants, and then followed by the artificial selection. Then we are going to select the better variety to produce a plant with the desirable trait of higher yield, nutrition, and resistant to disease that are going to be selected. So classical plant breeding involves this crossing, hybridization of pure line, and then there is a what a particular selection of the desired plants with the desired characteristic. So the new breed or a strain of animal or plant in which certain character appears in successive generation is the particular pure line. So what is pure line? Pure line are the homozygous plant, and these are produced by the continuous selfing, and due to which a certain character in a breed or a strain of animal and plant appear in a successive generation, then that is one of the important characters. With the advancement in the genetics, molecular biology, and tissue culture, with the increasing in this advancement in these three branches, plant breeding is now increasingly being carried out by molecular genetic tools. And what are these molecular genetic tools? We will study in the biotechnology chapter. If we were to list traits or characters that breeders have tried to incorporate into crop plant, if we talk about the, or if we list the traits or characters that breeders have tried to incorporate in the crop plants, the first we list would be increased crop yield and improved soil quality. So all the breeders initially talked about the crop yield and improved quality. Along with the time, the plant breeding was developed for the what crop yield and for the disease resistance, then increased tolerance to the environmental stresses like salinity, extreme temperature, drought, resistant to pathogen, insect resistance, and increased tolerance to insect pests would be on our list too. That also done in a sequence of time. Okay, so by understanding this many things, now what we are clear and what is plant breeding. If we answer that question, then we can answer that breeding is a process in which genotype of an organism is modified to make that organism process in which genotype of an organism modified to make that organism more useful to humans. If we do with this plant, then it is known as plant breeding. So if we describe this plant breeding is genetic improvement of crop plants in order to create desired plant type that are better suited for cultivation, give better yield, and are disease resistant. Okay, so plant breeding in this genetic improvement of crop will occur, and this will lead to the creation of a desired plant type which is better for the better suitable for the cultivation, can give a better yield, and that can be the disease resistant. Okay, now after this a new variety will be obtained. So what is a new variety? So it is known that a variety of group of plant consists of group of plant that have the similar genotype that have the similar genotype and it differs in one or more characters in one or more characters from other varieties of the same crop. Okay, so variety consists of group of plants that have the similar genotype and it differs in one or more characters from the other varieties of the same crop. Okay, it is somewhat modified than the other different varieties or species of the same crop. And a new trait or new character is established in the new variety. So a trait or character, if we explain, then a trait or character is any morphological, biochemical, or behavioral feature of an organism. So it is any morphological, biochemical, or behavioral feature of an organism. Like if we take example, yield of seed, size of fruit or seed, etc. Such character are the character, or we can say a trait. So we have discussed that many present day crops are the result of domestication, that is in the human use, and today all our major crops are derived from the domesticated animals. Okay, so this is all about the what we can say the particularly what is particularly a plant breeding. Now the question is we have to understand some of the terminology here that will help us to understand this chapter in a better way, and some terminology or terms that are important before studying the how the plant breeding is done. And there is a one term this is known as phenotype. If we talk of phenotype is an observable or external feature of an organism. Then second is genotype. Genotype is genetic makeup of an individual or a variety. So it is a genetic makeup is genotype. An external visible feature is a phenotype. Then the next term is line. A line consists of group of individuals related by descent and often similar in genotype. So individuals related by a group of individuals related by descent and often similar in genotype that will be constituted in a line. And the fourth is a pure line. It is a self-pollinated progeny. It is self-pollinated progeny of a homozygous plant. Okay, so that is the particularly which I can say the terminology. So pure line is what several descent. If four generation are there, the individuals of a related descent of one generation of one hierarchy, one heredity, these four generations will be considered as a line. But the self-pollinated progeny of a homozygous plant, and this homozygous plant is actually a pure line. Okay, so this is what we have understood now about the what is plant breeding, why the need is there, and what are the terminology. Now the question is how this breeding program is done, how the plant breeding is done. So plant breeding, if we talk about the plant breeding programs, if we talk, are carried out in a systematic way worldwide in government institutions, in commercial companies. Okay, and that plant breeding for the development of new varieties, these programs are carried in this systematic way. So if we talk about these steps, the main steps in breeding to have a new genetic variety of a crop are, means if we want to make any hybrid plant, and then we have to follow some steps, and here we are going to talk about the steps to create a new hybrid variety, how the plant breeding is done, how the new variety will be generated or new hybrid will be generated. That will be done by the following steps, and these following steps include the first step, that is a collection of variability. Means we have to collect a germplasm. Now what is germplasm, what is variability, that we will discuss in a continuation. The second, we are go have to elect this evaluate this variability, and we have to select the parent. Means we have to decide the parent here. So we will decide this parent according to the what a need of a character in the hybrid or the desired character. Then we will carry out a cross hybridization among the selected parent. First is variation selection, then parent evaluation and selection, then cross hybridization among parents. Then so many offspring will be obtained. Then out of which the particular offspring which having a desired characteristic will be considered as a superior recombinant. And here in the fourth step is selection and testing of superior recombinant. Then we will test, release, and commercialization of this new cultivar will be done. So these are the 5 steps which we are going to discuss here one by one in detail. So first we are going to discuss the collection of variability. So let's discuss collection of variability. So collection of variability, it is also known as germplasm collection. It is also known as germplasm collection. This germplasm collection or genetic variability is the root of any breeding program, and it is the first step. Okay, so it is one of the important step. So here, what is genetic variability? It is a tendency of individual genotype in a population to vary from one another. It is variability. Variability is a tendency of an individual genotype or genetic makeup in a population to vary from the one another. Means if we talk about a genetic of 100 individuals or 1,000 individuals in one population, the genetic makeup of every individual is going to vary. Now, in plants or in every organism, if we talk, generally pre-existing genetic variability is available from the wild relatives of the crop plants. Means wild is the original relative of the crop plant. Now mutation in occur in this, and the different particular new various species or various varieties will arise due to this mutation from the wild crop plant. So the collection and preservation of all the different wild varieties, species, and the relatives of a cultivated species, whatever the cultivated species is there, it's all the different wild varieties, species, and relatives, their collection, preservation, followed by evaluation for their characteristics. It is a prerequisite for the effective exploitation of natural genes available in the population. So if you want to exploit or if you have to use the natural gene which is present in the population, we have to collect all the varied varieties their relatives and their species. So it is a collection and preservation is important. So if we go and collect the entire plants, collection of an entire collection of the plants or seeds having all the diverse alleles for all the genes in a given crop is called germplasm collection. So what we are going to do in germplasm collection? We have to collect all the plant and seeds of all the diverse alleles. We have to collect all the plant species having all the diverse alleles for all the genes which are present. If we are considering taking a wheat crop, then we have to collect all its diverse alleles in all genes by collecting this plant and seeds, because we know that seeds have an embryo and embryo have all the genes of a particular plant which is going to be produced from that. So this way we are going to collect the seeds of them, so we can collect the germplasm. So when we collect this germplasm, what this germplasm consists of? So it is known that germplasm of any crop species would consist of following type of material, and this include cultivated, improved varieties. We have to collect the seeds of cultivated improved varieties. Then second, improved varieties, seeds of improved varieties that are no more in cultivation. Then third is old local or desi varieties. Fourth is the lines or descendants produced by plant breeders. And fifth is wild relatives related to crop species. Okay. Seeds of all these different varieties are going to be collected for the germplasm collection. And all these materials contain valuable alleles of genes that are important in the breeding. So this germplasm is a building material out of which improved varieties are going to be constructed or produced. In other words, we, we can say it is genetic variability or germplasm collection is the backbone or root of any breeding program, as we have discussed. Okay. Now it is known that this germplasm collection, it is one of the important point that germplasm collection are usually maintained at low temperature in form of cells. Okay, so that is the first step of the germplasm collection, and that is done by the collection of seeds of all the different alleles of all the genes. Now after collection of germplasm, suppose we have selected wheat crop and we have collected all the different varieties and all the alleles of all the genes. Now we are going to perform a second step, that is evaluation and selection of parent. Means out of this different varieties, hundreds of varieties, we are going to select a parent. Now, how we can select a parent? We are going to select a parent according to the our requirement of some specific desired characteristic in the hybrid, which is going produced by this parent. So now in this, what is done? The germplasm evaluated so as to identify plants with the desirable combination of character. It means I want to take a parent as a high having a high yield, and this is the reason that we have to search for the plant having a high yield, and the this is registered. So whole available germplasm is evaluated to identify plants with the desirable combination of the character. So we can select such plants as a parent. Suppose I am taking a longer grains of the rice as a one character, then I have to search whole germplasm for the seeds having a longer grains of rice. If I am going to do a breeding of a rice plant. Now in these, the selected plants are multiplied and used in the process of hybridization. Okay, and when these are multiplied and used in the process of hybridization, we can say two or more types of plants are selected which possess all the required traits among them. Okay, so two or more types of plants are selected, and this selected plant which having a two or more types of plant with the required traits among them, they are called parents. Now pure lines are created whenever desirable and possible of the particular parents we have selected. Okay, so in this we remember, out of the two type of parent, some plants are marked as female while others are marked as male. Okay, so we have to note two statements, that two or more types of plant are selected which possess all the required traits amongst them, and these selected plants are called parents. Then out of the two type of parent, out of two types of parents, some plants are marked as female while others are marked as male. And it is known that there is no such need of marking them as a male and female in case of unisexual plants. Now, the selected plants which are selected as a parent, this plant are multiplied and used in process of hybridization. As we have studied, pure lines are created whenever desirable and possible. Now, after the selection, evaluation and selection of parents, the third step is going to be carried out, that is a cross hybridization among selected parents. So in this third step, that is cross hybridization among the selected parent, we are going to perform hybridization. So the desired characters have often combined from the two different plants or two different parents. And for this combination of the desired characters, we have to make the hybridization. So this is the particularly A plant, and this is the B plant. So this plant have a flower, and this plant have a yellow flowers, and this plant have a blue flowers. Okay. Now when we make a hybrid, this have a both types of flower. So that is the how hybridization is taken into consideration, and hybridization is done, and the characters of both the desired plant, that is A and B, or parent plant A and B, is are getting expressed in the hybrid plant. So now, if we want to suppose need a high protein quality of one parent that may need to be combined with the desired disease resistance from the another plant. Suppose I want a particular new plant of a rice or wheat which has a high protein quality, and we can say disease resistance. Then have to select a one parent with the high protein quality and another parent with the disease resistance. So, like this, if I take and select a parent, it is possible by cross hybridizing the two parents to produce hybrid that genetically combine the desired characters in one plant, and that has both the character that is high protein quality and disease resistance. Such a way the hybridization can be carried out. So it is very time consuming process and tedious process if we consider. So here in hybridization, it is a process of making a cross between two genetically diverse parents to obtain a progeny with the desired superior trait. The desired character very often combine from the different parent, and we have taken this example as a high protein quality and its resistance. Now, to carry out this hybridization or cross hybridization, pollen grains from the desirable plant chosen as a male parent means if we have taken a male plant, if we have marked the high protein quality containing plant as a male plant, and the disease resistance containing having character having plant that as a female plant, then we will select we will select this pollen grain from the desired plants means a male plant that is having a high protein quality as a male parent, and its pollen grain have to be collected and placed on the sigma of the flower selected as female flower means the flower of the plant which has uh, we can say disease resistance. Such a way the pollen grain from the male parent is taken. Also, it is not necessary that hybrid do combine the desirable characters. It is also not necessary. Usually, it is considered that only one in few hundred to the thousand crosses show the desirable combination. Means if we are crossing a two parent plant and producing a hybrid plant, thousands of hybrid will be produced. Out of this, only one will have the desired characteristic. Like out of thousand, one plant we will obtain which has a high protein quality and the disease resistance. So this is the what a cross hybridization. Now the question is how this cross hybridization is done. Okay, so the cross hybridization is done here. And in cross hybridization, if we see the steps of this, its steps, the first is emasculation. In emasculation, it is essential. If the female parent bears bisexual flower, then the female parent, if female parent bear bisexual flower, then anther are removed, and this removal of anther from the bisexual female parent, it is known as emasculation. Then when anthers are removed, now the particularly bisexual flower has only female part, that is a carpel. So this female plant with the carpel is next bagged. So bagging is done. So bagging occur in which emasculated flower which do not contain anther or anther are removed is covered by a covered by bag of suitable size, generally made up of generally made up of butter paper, and they are covered to prevent contamination or interaction of its stigma with unwanted pollen. Okay, that is done in case of bagging. Now, when stigma of bagged flower attain receptivity, or we can say mature pollen grains are collected from the anthers of the desired male plant are dusted on the stigma. So first, after bagging, the third step is done, that is debagging, that is removal of bag from the emasculated flower when stigma of bagged flower attain receptivity. Okay, then after debagging, the desired pollen grain are dusted over this stigma because stigma is now exposed. So this next step is known as dusting, that is the mature pollen grain collected from anthers of desired male plant or male parent are dusted on stigma, and then fifth step is done, that is rebagging. Again bag is kept over this particular, so flowers after dusting are rebagged and allowed to develop. Okay, so this particularly rebag, and after rebagging it will carry out a pollen crystal interaction, and our crossing is done. So now we will mark that particular plant with the whose hybridization is done with the particular tag, and that is known as tagging. So it is to provide a label to the hybridized female parent. Okay, that its hybridization is done. Now, one of the important points that you need to know that if female parent produce unisexual flower, there is no need of there is no need of emasculation. Okay, so that is the thing which is done in the particular hybridization. Now here the parent can be of particularly the same species or same variety. So if we talk about the hybridization, the type of hybridization can be of several types. And if we talk about type hybridization, this hybridization between the two parents of same species or same genus can be done. So first is the type of hybridization are known as intravarietal. And this intravarietal is between two plants between two plants belonging to same variety. Belonging to same variety. And this between two plants belonging to the same variety that is useful for the self-pollinated crop means same variety. If I am taking a variety of tobacco, that is Nicotiana tabacum, then if I am crossing a Nicotiana tabacum with the Nicotiana tabacum, then it is known as the intravarietal. Then hybridization can be intervarietal or intraspecific. Okay, so we have taken example of Nicotiana tabacum, but its variety. Suppose I am taking a particular wheat variety, Sonalika, and if I am crossing the Sonalika with the Sonalika, then it is a intravarietal. Now we are going to talk about intervarietal or intraspecific. So in this, it is a hybridization between two varieties of same species. If hybridization is done between the two varieties of the same species, then it can be called as intraspecific within a species or between the varieties. And if we talk about the example, Ganga and Ranjit varieties of maize. If we make a cross between the Ganga and Ranjit varieties of the maize, that is intervarietal or intraspecific. Then after this, we can talk about the next type of hybridization. So now the third type of hybridization is between two different species, that is known as interspecific. So it is a hybridization between different species of same genus. And here the example are rice variety of ADT37, and this ADT37 is a rice variety which is obtained by a crossing of Oryza japonica and Oryza indices. Then after this, the next is intergeneric. Intergenetic is the hybridization between two different genera, or we can say genus. So if we take an example, that is triticale. Triticale is a hybridized plant of wheat and rye. Now wheat is Triticum testimum. So here genus is Triticum. And if we talk about rye, the rye is Cicale cereale. So this Cicale cereale is a rye. Okay, so that is the intergeneric. Another example, if we take, it is a Raphanobrassica. And Raphanobrassica is a intergeneric plant by the hybridization between radish and cabbage. This radish is Raphanus sativum, Raphanus sativus, and cabbage, that is a Brassica oleracea. So here you can see Raphanus and Brassica are the different genus. So it is a between the genus. So it is an intergeneric hybrid. Okay. So this is the third step, that is a cross hybridization. Now after the cross hybridization, we carry out this cross hybridization with the thousands of plant. And out of thousand, we have to select the superior recombinant. So next we are going to take about the selection and testing of superior recombinant, which has the desired characteristic. So it is considered that this step consists of selection among the progeny of the hybrids. And here those plants that have a desired combination will be considered the progeny of the hybrid. Now the selection process is considered as a crucial step for the success of the breeding objective. Okay, for this successfulness of this breeding program, or success of this breeding program, this is the crucial step. And this requires a careful scientific evaluation of the progeny. Okay, this step yields plants that are superior to both the parent. Okay, and it is known that these are self-pollinated for several generations till they come to the state of uniformity or homozygosity, so that the character will not segregate in the progeny, okay, or will not separate in the progeny, and they will remain together. So due to this, self-pollination is necessary. Okay, so now the parent is going to be superior. So here the term is used, that is hybrid vigor, or or we can say heterosis, and it is defined as superiority of parent over superiority of offspring over parent, sorry, means offsprings are better than the, or superior than the parent. So superiority of offspring over parents is known as hybrid vigor or heterosis. Now after the selection and testing of superior recombinant, next we are going to test this the superior recombinant, then we are going to release and commercialize this new variety or new cultivar. So the newly selected lines or the plant are evaluated for their yield, quality, disease and insect resistance, and for other traits before it is released as a variety for cultivation in a given region. Okay, so this newly selected line or newly selected plant is tested for this many things. like yield, quality, disease, insect resistance in the lab. So this is the disease and this is the resistance. Okay. So this evaluation is done by growing this in research field and recording their performance under ideal fertilized condition. Means we particularly evaluate them in the research fields. We grow in the research field, and in research field all the proper nutritions are there, proper fertilizers are there, proper irrigation is there, and proper crop management practices are in this. How this plant show its growth, that we are not going to evaluate. If it grow better, then after evaluation in the research field, the evaluation in research field is followed by a testing of the material in farmer field. And in farmer field or in agriculture field, it is done for the at least 3 growing seasons at several locations in the country. That is a different locations representing all agroclimatic zones. So which is going to these different growing seasons should represent the agroclimatic zones where the crop is usually grown. Now the material is evaluated and compared with the best available crop, local crop cultivar. Okay, then the material is going to be evaluated and compared with the best available local crop cultivar. When it is better, evaluated as a better new cultivar as a local considered as a better than the local variety, then finally the seeds of desirable plant are certified by NSC, that is National Seed Corporation, for marketing. Okay, finally it is done. Okay, and the evaluation in research field which was done earlier, that evaluation in research field is done in ICAR, that is Indian Council of Agricultural Research, New Delhi. Okay, so this way the whole particularly hybridization program is carried out. And finally, we have a new hybrid in the market. Okay, so now here, after understanding this, we have to note down some important points. Then we will talk about the green revolution in detail. So after discussing this breeding process, a technique of plant breeding, before talking about green revolution, let's talk about some crops and its type. Now in crops, there is a two type of crop first, that is primary crop. Sometimes you can get this question from this primary crops. So primary crops are the crop which under domestication from the very beginning. From the very beginning. And if you take example are wheat and rice. And another crop are known as secondary crops. The secondary crops is originally a wild relative of a wild relative of primary crop which was domesticated when primary crop failed. Example are rye and oat. So when primary crop failed, that time this secondary crop are used as a food, that is rye and oat. Apart from this, the another type of crop which are known as new world crop and old world crop. So if we talk about the new world crop, these are the crop of North and South America. And this new world crop include maize, potato, coca, tomato, cotton, red pepper, sunflower, cashew nut, peanut, cucurbits. These are new world crop. And the old world crop are the crop of Asia, Europe, Africa, and Australia. And this old world crop include wheat, rice, barley, pea, soybean, lentil, black pepper, cardamom, coriander, mango, sugarcane, tea, coffee, and almond. Okay, these all are the older crops. Okay, now after this, let's talk about the green revolution. And if we talk about the green revolution in more detail, dear student, we have already studied about the name of E. Borlaug. Dear students, we know that India is a mainly a agricultural country, and agriculture, if we talk, contribute or account for the approximately 33% of India's GDP. Okay, that is a gross domestic product. So 33% of the production or India's GDP. And it is known that this 33% of India's GDP is contributed by agriculture, and it gives employment nearly 62% of the population. So it is known that after India's independence, one of the main challenges faces the country was that producing enough food for the increasing population. Now we know that as only limited land is to fit for cultivation, only limited land is available or fit for the cultivation. So India has to strive to increase the yield or need to focus on the area to increase the yield per unit area in existing farmland. So for that, the development of several high yielding varieties of wheat and rice in the mid 1960s was done. And a result of improvement or development of these high yielding wheat and rice varieties, various plant breeding techniques led to a dramatic increase in the food production in our country. And this phase of mid 1960s is referred as a green revolution. So now, following this green revolution, some Indian hybrid cross high yielding varieties was produced in which wheat, maize and garden pea was produced. Now if we talk about this wheat and rice during the period of 1960s to 2000, wheat production increased from 11 million ton to the 75 million ton, while rice production went up from 35 million ton to 89.5 million ton. It is not 889, it is 89. So this much improvement was done. So this was due to the development of semi-dwarf varieties. Okay. So this was due to the development of semi-dwarf varieties of wheat and rice. So now we are going to talk about this varieties, how these varieties are developed. Now it is known that Norman E. Borlaug at the International Center of Wheat and Maize Improvement in Mexico developed this semi-dwarf wheat. So if we talk about the semi-dwarf wheat or a dwarf wheat, the gene responsible for dwarfing in wheat is gene responsible for dwarfing in wheat is Norin 10. Okay, and this Norin 10 gene was reported in Japan. Now it is known that this variety is developed by Norman E. Borlaug at the Mexico. Now this these varieties or semi-dwarf varieties developed at Mexico had high yield, resistance to lodging, common pathogen, and pests. These varieties are fertilizer responsive, they grow better in the fertilize the soil, and they have smaller growth period. Okay, so these varieties which are developed at Japan, these varieties which are developed in Mexico by the Borlaug has these many characteristics. Now the MS Swaminathan, who was the Indian scientist, this MS Swaminathan initiated collaboration with Dr. Borlaug, which culminated into the Green Revolution through introduction of Mexican varieties of wheat in India. So when Mexican variety is introduced in India by the collaboration of Borlaug and MS Swaminathan, this enhanced the Green Revolution. Now it is known that Sonora 64 and Lerma Rosso 64, these are the two important varieties of wheat that were brought to India and modified through gamma radiation. Now there is a need of this particular modification by gamma radiation because only by this mutation they can adjust in Indian environment and they become a part of Indian agriculture. So they become part of Indian agriculture. Now it is known that in 1963, so after this, in 1963, several varieties such as Kalyan Sona and Sonalika, which are high yielding disease resistant varieties, were introduced all over the wheat growing belts of India. Okay, so that is the dwarf wheat varieties which was introduced introduced in India, that is responsible for one of the and it is one of the reason for the green revolution. Apart from this, the dwarf wheat, we can say dwarf rice, semi-dwarf varieties of rice also developed. Now it is known that gene responsible for dwarfing in rice is DGO Wu gen. Okay, and this gene was reported in Japan. Now it is known that it was this gene was introduced in rice varieties by International Rice Research Institute, Philippines, in varieties like IR8, IR24. And it is known that these are the Norman E. Borlaug and the MS Swaminathan. Now, this Swamidar rice variety were derived from the IR8 developed at International Rice Research Institute, Philippines, and Taichung Knitting 1. Okay, from these two varieties, a modification which these two varieties, that is IR8 and Taichung Knitting 1, developed at the Taiwan. Now, modification is this done and the semi-dwarf rice varieties were developed. Now, this derivatives were introduced in 1966, and it is known that this IR8, it later on the better yielding semi-dwarf rice varieties like Jaya and Ratna were developed in India. Okay, so this is the what a dwarf rice. Now after this we are going to talk about the sugar, and in sugar cane two varieties are there. One is sugar saccharum barberi and another one is saccharum officina. Now if we talk about the saccharum barberi, It was originally grown in North India but had a poor sugar content in yield. We remember it is grown in which one? North India. But it had a poor sugar content and yield. Now that is need to be improved. Then it will be the better quality. And in tropical cranes, which is grown in South India, that is the Saccharum officinarum. This had a thicker stem and higher sugar end, but it did not grow well in the North India. It is not grow in North India. And the North Indian variety, that is a Karam barberry, that had a poor sugar content and yield, but it has a higher sugar content and thick stem. So if we cross them and we make a hybrid, then it is possible that the next offspring will have a better yield. We can say better yield and high uh, sugar content, and as well as it will be able to grow in the North India. So this is the Saccharum officinarum. Now these two species were successfully crossed to get the sugarcane variety combining the desirable qualities of high yield, thicker stem, high sugar, and ability to grow in sugarcane areas in the North India. And it was happened. This hybrid has this characteristic. Okay, so this is the way how the sugarcane hybridization is done. Now, after this, the millets were also studied, and it is known that the plant producing a large crop of small seeds are known as millets. So millets are plants producing a large crop of small seeds. They are called as millets. Now hybrid maize, jowar and bajra have been successfully developed in India. Hybrid bajra, hybrid jowar, hybrid maize were developed successfully in India. Now hybrid breeding led to the development of several high yielding varieties which are resistant to the water state, and such a way the maize, jowar and bajra was developed in the millets. Okay, so this is all about the what green revolution and the plant breeding technique. So green refers to the increased production in agriculture. Obviously, father of green revolution in world is obviously Norman E. Borlaug. If we talk about India, then we can call MS Swaminathan. The entire collection of plant or seeds having all the diverse alleles for a gene in the given crop, it is nothing but a germplasm collection. So answer will be 3. Okay, so this is all about the what a plant breeding technique, what is plant breeding, and how the semi-dwarf variety is developed, and how they are responsible for the tree production. That is all done. I hope you understood well. Now the next topic we will continue in the next lecture. Till that, thank you everyone.